This is chapter 13. We continue our discussion about genetics and biology, but now we're going to be looking at molecular genetics, digging into the molecule that contains the hereditary information we've been talking about in the recent chapter with Gregor Mendel and other types of inheritance patterns. So to give you just a little preview, we're going to be learning about a little bit of the history first about how DNA came to be discovered as that molecule of heredity. And then we're going to talk about the structure of DNA and how DNA can replicate itself. So the first part of this lesson, and probably just this lesson, this video is going to focus on some of the history about how DNA came to be discovered. And let's pick up where we left off in our last unit, just as a recap. Remember the work done by Thomas Hunt Morgan on his fruit flies, and he came up with the chromosome theory of inheritance when he discovered that certain genes are located on certain chromosomes. Specifically, he determined that the allele for eye color in fruit flies is located on the X chromosome. Once Morgan and his group of scientists showed that genes are located on chromosomes, the two constituents of chromosomes, which are proteins and DNA, were thought to be the candidates for the genetic material. Nobody really knew, though, what that molecule of heredity was. So it was really a toss-up between DNA or proteins that was responsible for passing information from parent to offspring. So there were other scientists working on this and doing various experiments looking at organisms like bacteria and viruses. And so we're going to look at some of those now that led to the discovery that DNA is indeed the molecule of heredity. Here's one of the earliest studies in that regard where Frederick Griffith, in 1928, he was studying the bacteria that causes pneumonia in mammals. It's called Streptococcus pneumoniae. Perhaps you've heard of it. And he was working with mice and subjecting them to different treatments with this Streptococcus pneumoniae, whether it was the strain that could actually cause pneumonia, giving the mouse pneumonia, which ultimately would kill it, or whether it was not going to cause that effect because it was heat killed. Basically, that means it's superheated, so the bacteria are not active, they're not alive, they can't actually transmit the disease. However, the components that make them up, they're still there in that mixture that's being injected into the mice. It's just they're dead. So let's take a look at the 4 different types of trials that Griffith did that led to a pretty cool discovery about how organisms can transmit genetic information. So in the first experiment, if you look at the first row here, they have the living virulent or S strain. So that again means that that particular concoction would cause pneumonia and did indeed, when injected into the mice, killed them. They got pneumonia, they died. Okay, so that's kind of like a control to show that that strain is active and going to cause the disease. So then to show that the heat killing of the bacteria would be effective and that the mice wouldn't transmit pneumonia, they injected them with that was later. Let me hop down to number 3 since I was thinking about that. They took that same living virulent strain and they basically superheated it, killing the bacteria, but they still injected into the mice and the mice lived. So that shows that heat killing is effective at rendering the bacteria from not being able to give the disease to the mice. They didn't get pneumonia. Okay, so they worked with another strain of Streptococcus pneumoniae, the R strain, which is also a living strain, but it's avirulent, meaning that it's harmless. It doesn't cause the pneumonia to occur in the organism that's injected to. And so that was shown here with the red, the living avirulent strain. When injected into the mice lived. And So those three first trials are really just control experiments to show what they thought was supposed to happen. Then they started to get creative. They mixed together the living avirulent strain and the heat-killed virulent strain. And even though both of these, when separate, didn't kill the mice, if you take a look up here, we're looking at the second row and the third row. Independently, that did not have any effect on the mice. They lived. But when mixed together and injected into the mice, they died of pneumonia. How is that possible? Well, I hope you pondered it for a moment. From the mice that died, they were able to recover pathogenic bacteria from the mice blood. So that tells you that the mice died of pneumonia and the bacteria that were still there in their body had pneumonia. Yet neither of these alone were able to transmit pneumonia. So they discovered that there's this process of transformation that occurs where the living avirulent strain were able to take some of the genetic information from the heat-killed virulent strain, the gene that codes for the ability to transmit the pneumonia. They took it and they incorporated it into their own genome. And then, as such, they became now living virulent. So bacteria are one type of organism that have this ability to do this, to take bits of DNA from outside their own body and incorporate it into their own genome. And that's actually something that we get to do in our lab when we do genetic engineering. So this was a pretty pivotal experiment that led to some other research because still at this time, nobody really knew what the substance, what the chemical compound was that was responsible for this transformation. So another group of scientists, a few years later in 1944, were continuing the work of Griffith, and they isolated the different organic components from the pneumonia-causing bacteria. So they basically took the bacteria and they put them in a scientific blender, if you will, to get all of the cell parts into a homogeneous mixture. And then they put it in a centrifuge, which, spinning at really high speed, separates out the components into layers, and then they can isolate out the different components as proteins, RNA, DNA, carbohydrates, lipids. And then they basically did the same kind of experiment as Griffith. They took the virulent, so they did this with the virulent S strain. They isolated and purified all of those components, and then they mixed that with the avirulent R strain, and they looked to see which of the mixtures would cause the mice to have pneumonia. And the only one that did was the, come on, highlighter, DNA. DNA from the virulent S strain, when mixed together with the avirulent R strain that was alive, of course, just the DNA of the virulent strain isn't like a live bacteria, it's just part of it. But that shows that it was able to incorporate it into its own body and then become virulent and transmit pneumonia to the mice. Wow, what a finding. How do they think of this stuff? It's just fascinating, I think. Now, any good research requires replication or further study to just verify those findings. So, of course, there were still other scientists around this same time working towards finding what was that molecule that is responsible for transformation, for heredity. And so to further the evidence that DNA was the genetic material, there was another group working with viruses. And viruses that are able to interact with bacteria. And so how a virus works This is one example of a virus called a bacteriophage, or a phage for short, Phages. It injects its own DNA into a host cell, and the host is where replication occurs for the virus. So the virus itself is a very simple, it's not really an organism, simple thing. It's made of two main components, a protein coat, or protein body, if you will, shield, that protects the sensitive DNA inside. For this particular one, this is a DNA. There are some viruses that are RNA based, but this one's DNA based. So they knew that the bacteriophage was mainly composed of protein and DNA. And if you recall, those are the two chemical compounds that were being looked at as which one is the molecule of heredity. Is it the DNA or is it the protein? So this group did an experiment using radioactive labels for the protein and the DNA to see which of those would be transmitted into the bacteria where then replication would occur. So in the first experiment on the left, they used a label for the protein coat. They used sulfur, because sulfur is only found in protein and not in DNA. So they labeled it with a chemical that kind of fluoresces and can be examined in the lab. They grew the culture with the radioactive sulfur. That's what they really did. Okay, but this is like a zoom in of what they would look like. And then they infected the bacteria. So they added the bacteria to the virus culture. And, I'm not sure how long that sat, but long enough for them to know that if they were going to infect them, that they would. And then they ran a centrifuge to separate out the layers. In this tube would be, at the top, would be the viral particles, because they're going to be smaller and lighter than the bacteria. The bacteria would be at the bottom in what's called the pellet. Since the radioactivity remained in the liquid above the pellet, which is called the supernatant, and that would be where the viruses are, indicates that the radioactive components, which were sulfur on the protein coat, were not transmitted to the bacteria, since the bacterial part of this separated layers remained non-fluoresced. Okay, so that just proved that proteins are not the genetic material, but they still have to prove that then DNA is. So they did a similar experiment, but except this time they used a radioactive label for phosphorus, which is found in the sugar phosphate backbone of DNA, but not in protein. So basically they did the same thing. They put the phage they grew the phage culture with the radioactive phosphorus, then they added the bacteria, let the phage infect the bacteria, centrifuge the cultures, and look for the separation of the bacteriophage from the bacteria. And this time the radioactivity went in the pellet where the bacteria is found, which indicates that the genetic information is the DNA because it was transmitted from the virus into the bacteria where it remained in the final tubes after centrifugation. So to conclude, they found that DNA is indeed the genetic material. Awesome. Okay, so now in our next lesson, we're going to learn more about the structure of DNA from the next sets of studies that led us to learn about both the chemical structure and the physical structure of DNA.